Hello, good afternoon. My name is Ogechukwu Etuazim, and I worked with the O'Neill Lab. And our presentation is Spatial Transcriptomics, the Regional Importance in the Female Reproductive System. So examining gene expression. So there are a lot of modes of examining gene expressions, but we're only going to be focusing on a few today. So we have a targeted method, also known as qPCR, which targets a specific gene in order to see the expression of that particular gene. Then we have untargeted expression, which is also known as unbiased single-celled RNA sequencing. It detects all of the genes that are expressed in a cell. Some of its limitations include that you would have to use all of the tissue sample, and you would lose some of the information about the tissue's architecture and regionality. So a newer method of examining gene expression is known as spatial transcriptomics. Spatial transcriptomics is a molecular profiling method that allows scientists to measure all gene activity in a tissue sample and map where the activity occurs. So why is location important in the female reproductive system? So this is an image of the female reproductive system. It consists of the ovary, fallopian tube, uterus, endometrium, cervix, and the vagina. So follow along with me as I give you 3 examples as to why location matters. So the first organ we're going to look at is the ovary in regards to the oocyte development. So one of the most important roles of the ovary is that it's the location where the egg or the oocyte develops. Oocytes must undergo a growth and development process within the ovary before they are capable of being ovulated and fertilized by the cell. So there's actually a unique physical distribution of follicles within the ovary, with the vast majority of small resting follicles that are located in the outer portion of the cortex, outer region of the cortex, while the growing and maturing follicles are in the inner portion of the medullary. So we don't know exactly why that happens, why the follicular migration happens. So some questions that we hope to answer with spatial transcriptomics include what drives the migration of the follicles? Is it driven by the oocyte, the follicles, or factors that are secreted by cells in the cortex or medulla? So we would need to analyze the oocytes and the surrounding niches using spatial transcriptomic data to be able to answer those questions. The next organ we'll be looking at is the fallopian tube regarding ectopic pregnancies. So ectopic pregnancies are pregnancies that develop outside of the uterus that leads to the embryo not being able to develop to full-term pregnancies. Ectopic pregnancies are most commonly located in the ampular portion of the tube, and ectopic pregnancies can rupture and can be life-threatening. About 2% of women experience ectopic pregnancies. So here is an image of the female reproductive system. So if an embryo does not implant itself within the uterus and implants outside of the uterus, it's known as an ectopic pregnancy. What we found is that when it implants in the fallopian tube area, most of it, 70% of it implants in the ampullary region. If it doesn't implant there, 12% is in the isthmic, then 11% in the fibril, 3% in the ovarian, and 2 to 3% in the corneal region. So we currently don't know exactly what is driving those ectopic pregnancies to happen. So we're hoping that with more data and more use of spatial transcriptomic data, we will be able to determine the driving force of the genes that lead to those ectopic pregnancies. And the next organ we'll be focusing on is our uterus and early pregnancies. So the uterus is the womb in which the fetus develops. In order for pregnancy to occur, an embryo must attach to the endometrium, which is the innermost layer of the uterus. Implantation typically occurs to the posterior portion as opposed to the anterior portion of the uterus. So here is an image of the uterus, and here is the endometrium that consists of the stratum basalis and the stratum functionalis. So usually what we typically see is that implantation occurs in the posterior portion as opposed to the anterior portion. However, we don't know exactly what drives for the embryo to implant in the posterior portion. So we're hoping that with further research using transcriptomic data, we would be able to tell what genes, maybe if it's a gene or anything that drives the implantation in that manner. So some of the challenges that we face with using spatial transcriptomic data is, for one, the cost. It's actually very expensive. It's about $40,000 to $43,000 to use spatial transcriptomic data. Um, Resolution. So right now, technology hasn't been perfected to where we can just simply see into like single cell. We can mostly just see into like clusters of cells. And then compatibility with archival tissue. So that's simply just referring to how the tissue is stored. So it can either be stored with fresh frozen or FFPE. FFPE just meaning formalin fixed parafilm embedded. So FFPE is actually not very compatible with spatial transcriptomics, just with the fact that since it's been embedded, we would have a hard time being able to like go into the cell and look into it without being able to like break like the disulfide bonds to be able to see it properly. So I would like to extend a special thanks and acknowledgement to the O'Neill Lab, Dr. Kate O'Neill, my PI, Addie Johnston, the research specialist. If it wasn't for them, I don't think I would have been able to get through the presentation. I learned a lot and they taught me just a lot about the female reproductive system. I would also like to thank the NIH, Dr. Zarina Gallus, Dr. Ajay Pillai, and Julie Kim, because they were able to like mentor us and how to properly, do a presentation. And I'd also like to thank HuBMAP Consortium. And if anyone has any questions to me, for me, I'll be glad to answer them. Thank you.